Welcome. Starting with very basic and important question. What is the purpose of fumigation or fogging in pharmaceutical facilities? Fumigation or fogging is done to reduce microbial load on exposed surfaces and air, especially in controlled areas. It supports environmental controls required under UGMP Part 1, Chapter 3 and 5, Cleanliness and Contamination Control. What is frequency for fumigation in the pharmaceutical industry? Fumigation or fogging is typically performed on a routine schedule, that is twice in a month, or after a major shutdown modification, or if environmental monitoring shows microbial counts exceeding limits. Which chemicals are used for area fumigation in the pharmaceutical industry? The current preferred methods uses a dry fog generated from solutions of hydrogen peroxide and silver. For example, Virusil, Bramisil and Silvicil. These are non-toxic, eco-friendly and decompose into water and oxygen, leaving no residue. Which were traditional methods for fumigation? Traditionally, a reaction between formaldehyde solution and potassium permanganate was used. This method is now considered illegal in many regions due to the carcinogenic properties of formaldehyde and associated health risk for personnel. What are preparation steps for fumigation? Step 1. Stop all production operations and remove all materials from the area. Any remaining items must be covered with polythene bag. Step 2. Ensure the area is thoroughly cleaned before fumigation, as the process is disinfection method, not a cleaning one. Step 3. Switch off the air handling units, that is AHU, and adjust them to a specific fumigation mode and seal all windows and doors. What is contact time and defumigation method? Allow the fog or fumes to remain in the area for a specified contact time, typically 1 to 7 hours, to ensure efficacy against microorganisms. After the contact time, the area must be defumigated by running the AHUs continuously for a period, for example 30 minutes, to clear the air of any residual agent. What is post-fumigation procedure? After defumigation, clean the area and external surfaces of equipment using 70% volume by volume isopropyl alcohol, that is IPA, and a lint-free cloth. Very frequently asked question: what is the difference between fumigation and fogging in a pharmaceutical context? While often used interchangeably, fumigation traditionally refers to using gaseous agents like formaldehyde. Fogging is the modern preferred method that uses non-toxic, eco-friendly liquid solutions such as hydrogen peroxide and silver ion solutions dispersed as a fine mist or dry fog. Is fumigation mandatory as per GMP guidelines? No guideline explicitly mandates fumigation, but UGMP Annex 1, that is Contamination Control Strategies, that is CCS document, requires effective microbial control. Fumigation or fogging is considered a risk-based supplemental method. What is the recommended concentration of H2O2, that is hydrogen peroxide, for fogging? Typically 6 to 12% weight by volume for surface sanitation via fogging. The exact concentration must be justified during validation. Should HVAC, that is heating, ventilation and air conditioning system, be on or off during fumigation? Normally off during fumigation and on during aeration. This is determined during PQ and based on the CCS, that is Contamination Control Strategy. UGMP Annex 1 requires documented parameters for cleaning and disinfection. Very important question: what parameters should be controlled during fogging? Chemical concentration, volume of disinfectant, fog volume output, temperature and humidity, area cubic volume, exposure time. These parameters must be defined as a part of PQ. What are the 4 key stages of a typical vaporized hydrogen peroxide, that is VHP decontamination cycle? Stage 1 conditioning, Stage 2 gassing or decontamination, Stage 3 aeration, and Stage 4 post-cycle ventilation. This cycle is described in detail in manufacturer technical files and is the basis for validation as per PDS technical report number 51. What is a biological indicator or BI and why is it critical for validation? A biological indicator is a standardized High population preparation of a specific microorganism, for example, Geobacillus stereothermophilus spores for VHP, placed in a hardest to reach location. Its complete inactivation demonstrates that the process delivers a sufficient uniform lethal dose. Reference UGMP Annex 1. What type of environmental monitoring tests are done after fogging? Contact plates, swab sample, second plates. Annex 1 requires verification of disinfection effectiveness as a part of EM, that is environmental monitoring program. Very important question: what are common failures observed during fogging validation? Uneven fog distribution, insufficient concentration, non-sealed areas, equipment not cleaned before fogging, and environmental monitoring failure post-fogging. Annex 1 requires deviations to be investigated. What are acceptance criteria for fogging validation? 3 or more than 3 log detection for vegetative bacteria, 2 or more than 2 log detection for fungi, 0 growth and saturation hotspots as per risk assessment based on EN 13697 and EN 12